Good morning, everyone. My name is Sara Bell. I'm an analytical chemistry student within the Sweetler Lab. And my role in the Sweetler Lab is in small volume and single cell mass spectrometry. And today I'll be talking about a project that's different from the one that Martha introduced earlier today. This one is about the single cell chemistry of the Stickleback fish Pituitary and the implications in parenting behaviors. So this is an MBM-supported collaboration with Allison Bell's lab, and the Bell lab in particular is interested in understanding the parenting behaviors of the three-spined stickleback fish. So these fish are interesting because their parenting behaviors can vary differently from population to population, both within populations, so fish that live together in the same area, and across different populations across the world. So in particular, we're interested in two ecotypes from these fish, the common ecotype and the white ecotype. So these two fish are interesting because their parenting behaviors are the exact opposite. And for these fish, the males are the main caregivers. So in the case of the common male, he is an attentive father. He will take care of the eggs that are laid in his nest. He will clean them, oxygenate them, and then even take care of the young for a few weeks after they hatch. Now, on the other hand, the white male is an inattentive father. He doesn't really take care of his eggs. The second they're laid in his nest, he actually scoops them up in his mouth and just spits them out into the water so they can fend for themselves. So with the Bell Lab, we're interested in understanding how such different behaviors can occur within fish from the same species. So one step towards that the Bell Lab has worked on previously is looking at some transcriptomics data on just the common fish. So from that, they did show some differential expression of genes during different phases of parenting behaviors. Now, they're interested in furthering that type of data and looking at a comparison between the common and the white. And to kind of complement the directions that they're looking in, the Sweetler Lab, we want to bring in our single cell mass spec specialty to look at what peptides, proteins, and hormones are being used within the brains of these fish. So to do that, we use single cell mass spectrometry, and the way we do this is pretty specialized to our lab. So what we do is we collect fish from both of the ecotypes, collect their brains, and in this case, I'm using a brain adjacent structure, the pituitary, and we collect that pituitary then, and then break it down into its cellular components. Those cells are then placed on a microscope slide and dyed for fluorescence microscopy. From that microscopy, I get an image of all of the cells on the entire slide surface in relation to a bunch of expoducial markers around all the edges of that slide. So using that image and a program that was built within our lab called MicroMS here, this allows me to actually locate all of the individual, well-localized and separated clean cells on the slide. So once I've located those cells using their circularity, size, and the distance between them to filter, MicroMS will actually give me a pixel coordinate of each cell, which I can then translate to a physical coordinate on the actual slide when it's placed within our mass spec. So once it's placed in there, MicroMS will do that coordinate location for me and then give me that list of coordinates, which I can then use to direct a multi-laser directly at each individual cell and then ionize its chemical components to then collect mass spectra for each individual cell. Now from that, I've done MultiMS of about 40,000 cells at this point across both ecotypes. And for every cell, I get a mass spectrum. Every mass spectra, I get a list of what peptides we detect in each cell. So using those lists, there's a couple of things that we want to do. The first is, of course, identify what peptides and their respective proteins and hormones we're seeing. So I use a peptide library that was built in our lab using LCMS data of the pituitaries from these fish. And then once we can figure out what those peptides are, we want to ask are certain peptides characteristic of one ecotype or the other. So to do this, we use machine learning classification. With such a large data set, we have to use something to deal with that. And using this machine learning model, we can train it to either classify or categorize cells based on their chemical components. So from that, we get this UMAP visualization here, which just tells us which cells are chemically similar. So any of the cells within each cluster, take for example this purple cluster, those cells are all more chemically similar to each other than they are to any cells in any of those other clusters. And that goes for all of the cells within each cluster, and for each of those clusters, we've basically established a cell type that's based on the mass spectral chemical profile. So now that we have these chemical profiles established, we then want to see if any of those correspond to one ecotype or the other. So we can overlay that ecotype information, common versus white, and take, for example, this middle orange cluster. You can kind of see that it seems to be enriched in the white fish cells. And so we want to see if that average chemical profile of an orange cell can be related back to the behaviors of the white fish. So we've started to look into that, and we do see that certain peptides are enriched or characteristic of common cells versus white cells. So for the common fish, we see multiple peptides from corticotropin releasing hormone, and for the white fish, we see peptides from proopioid melanocortin, melanin concentrating hormone, and prolactin. Now, if you're familiar with any literature about parenting behaviors and the hormones involved in that, you may be wondering why we're seeing prolactin, a hormone that's associated with parenting behaviors, in the non-parenting fish, the white fish. Well, that's one of the questions that we're looking to answer moving forward. So my current directions with this project is actually being able to work with fish that are showing the interesting parenting behaviors. Sadly, the last fish that we got and all the data that I've shown you was for fish that were in a territorial phase, which is actually before any parenting behaviors occur. Now we've been able to get fish, been able to actually get them to start nesting, doing those interesting parenting behaviors, and we're moving on to do that same mass spec and machine learning analysis to then compare their peptide profiles when we see these interesting behaviors. Now, kind of a secondary direction that we'd like to look at, the Bell Lab is looking into characterizing some hormone receptor counts for hormones of interest, just to give us a little more context as to the usage of these within the brains of these fish. And with that, I'd like to thank everybody from the Sweetler Lab and Bell Lab that have been involved with this project, and thank the MBN program for its support. Take any questions. Yeah.
More than a question, I think it's just an interesting point that genetic differences and how you express proteins, that the cells express proteins, could affect human behavior too. And more than psychology, more than like what's good, what's evil, right?
Yeah.
Can it be determined by protein expression?
I mean, that's kind of the interesting thing about this. Obviously, we're working with fish, but parenting behaviors are pretty universal across different species. So yeah, kind of the same idea that you're picking out there. All right, any more questions? Thank you.